Today we're going to talk about the digestive system. That we have the digestive system divided in two divisions. The first division is the gastrointestinal tract itself, and the second division, digestive system 2, will be on accessory digestive organs. In part 1, digestive system 1, we have three parts to it. We have part 1 has to do with the cells of the digestive system. Part 2 has to do with the oral cavity, esophagus and stomach, and then part 3 has to do with the small and large intestine. Digestive System 1, Part 3, Small and Large Intestine. With the Digestive System 1, we want to look at the parts of the digestive system, the 4 different layers on the wall. We want to know and identify distinguishing structural features within different regions of the digestive system, as well as identify the cell types. Part 1 has to do with cells, Part 2 has to do with the oral cavity, esophagus and stomach, and Part 3 will finish digestive system 1, which will be the small and large intestine. Now, when we're coming out of the stomach, the pyloric region, we see the pyloric glands. These are pyloric glands right here. Note that they're in the lamina propria. There's a muscularis mucosa right there. There's a pyloric sphincter. A pyloric sphincter that regulates the flow of fluid from the stomach into the small intestines. So here we have gastric pits in the stomach. Then all of a sudden we have projections on the surface. With the pits, we also have intestinal glands. But in addition to that, these villi project on the surface. These are small intestinal villi, as you have no villi in the large intestine. In the submucosa, the muscularis mucosa kind of broken up in through here, but in the submucosa is where we have these duodenal glands or glands BRUNNER, muscularis mucosa right here, epithelium, lamina propria, that we are seeing. So in the pyloric stomach, we have these glands in the lamina propria. In through there, these are gastric pits, submucosa, muscularis externa, and you have a serosa, which is a layer of cells, mesothelial cells on top of connective tissue. Here in this slide, we see the pyloric duodenal junction. So here we get the this is the pyloric region, and here, starting here with these glands, the Brunner's glands that are in the submucosa, right in through there would be the junction between the stomach and the small intestines with the villi. They're kind of fragmented here, but those are the intestinal villi projecting off the surface. Now, the small intestine organization are, again, here's the stomach, here's the small intestines. These have gastric pits that we see there. In contrast, small intestines have these crypts of lubricant. So these are the intestinal glands that project down here that correspond to the pits. There's also epithelium is a little different in here. You have absorptive cells where you did not have those in the stomach. We also have endoendocrine cells or endocrine cells. Argentaffin cells is another name for them, and we actually have both of those, have those in the stomach as well as the small intestines. But other two cell types we do not have in the stomach. One is the Paneth cells, which sit right at the base of these intestinal crypts of lubricant, and then we have goblet cells, and we can see the light staining cells in the pink epithelium there, goblet cells. No goblet cells in the stomach. Here in the duodenum, you see the mucosa, submucosa, located here, the muscularis mucosa down through here with the submucosal glands of Brunner, located right in through there. And we see a lot of those. You can see how they are discharged from their location through these intestinal glands and to gain access to the surface. You can see a hieroglyph of that here, where you got the Brunner's glands empty into the glands of the intestine, the crypts of lubricant. Here you can see another one where these type cells are giving rise to connecting to the lumen. Now, in the small intestine, there's about 3 things that amplify the surface. One is the pachycircularies, which are kind of long folds that we have. We have a submucosa increasing. And then in addition to that, you have these projections off of these, which are the intestinal villi. And you see a single intestinal villus here covered by epithelial cells. A simple columnar epithelium has goblet cells and absorptive cells, Argentaffin cells, and through there, even some pennant cells located at the base here in these intestinal glands. In the connective tissue, you have blood vessels going in and out, of course, but you also have a blind ending lymphatic. This is the centrolacteal. So we have amplification by infolding of the submucosa, projections of the villi. In addition to that, you have the intestinal absorptive cells amplify their surface area by microvilli. So those are the 3 things that amplify the surface, as we'll talk about here in a little bit. Here's a pyloric stomach. Again, note that the glands are in the lamina propria as opposed to the duodenum. They're located in the submucosa. There's a muscularis mucosa. There's a mucosa from there to there. Epithelium, lamina propria, muscularis externa. Here we can see the mucosa, which is a mucosa epithelium plus the muscularis mucosa and the lamina propria. Those 3 things, epithelium, lamina propria, muscularis mucosa, make the mucosa. And below the mucosa, you've got the submucosa, and that's where you see the glands. Glands located in through there, and this is the muscularis externa with a little bit of serosa maybe here on the end. Down in through there, you will see these pennant cells, which have the granules up above the nucleus to be discharged in the lumen. Also, there's these endocrine cells who actually touch the surface, but the granules in the base. Being endocrine, they discharge their content in the lamina propria and hence in the bloodstream for distribution throughout the body. So intestinal villus, as you can see projected up here, has connective tissue core, blood vessels in through there, as well as centrolacteal, and it has intestinal absorptive cells, simple columnar epithelium with a brush border in through there. We can see some lymphocytes migrating through, but that's what we see. And then these are the glands of Brunner, a mucus type secretion that helps to raise the pH from ingesta coming from the stomach. And here we can see again where the submucosal glands occur in the duodenum. You do not find that in other places in the small intestine. And here we can see the intestinal villi as well as the pachycircularies, the infolding of the submucosa, which amplifies the surface. Here we see in the duodenum, we can see the intestinal villi projecting in through there, and there would be the intestinal glands right in through here. You can see the intestinal villi, these projections, which has epithelium, lamina propria in the core and at the base, and then finally you have the muscularis mucosa and then the submucosa. If you have a special stain to be able to see the Argentaffin cells, here we can see this is an Argentaffin cell with its granules at the base. There it is again for discharge, and this is a contrast to the pennant cells. There's this nucleus and its granules are projecting up to the lumen because these cells, their granules are capable of killing bacteria, and so they're sitting here just waiting for bacteria to come down through the system. So here's a jejunum. There's a duodenum in through here and a jejunum kind of in through here. And we can see the muscularis externa, submucosa, lamina propria, and the lamina propria, muscularis mucosa, and the epithelium on the surface with the intestinal absorptive cells as well. Also, right in through there is a serosa. You can't see it very well, but you can see the serosa right here, connective tissue with a simple layer of mesothelium, kind of squamous cells. Sometimes they're cuboidal, but they're kind of squamous cells, simple squamous cells, mesothelium, which line the outside of the tube of the GI tract. Also, we have the pachycircularies. Remember, we had the infolding of lamina propria, and here we see muscularis mucosa kind of infolding of lamina propria to amplify its surface. We have the central lacteal in the center of the intestinal villus, epithelium, goblet cells that we see brush border on the surface. At the bottom of the intestinal glands, we see the Pennant cells. The Pennant cells located here with big granules that we see oftentimes are very orange granules. Also, we see in the muscularis externa the intrinsic plexus, the Auerbach's plexus. You can see the snake eyes of the nuclei with the nucleolus nissal substance here of the ganglion cells. Also in the submucosa, you can have a plexus there. That's the Meissner's plexus that regulates contractions of the muscularis mucosa. So in the small intestines, we have intestinal absorptive cells located in through there, and those intestinal absorptive cells have a brush border. Brush border is uniform microvilli of similar thickness, and they also have the glycocalyx. In between adjacent cells, you have junctions. You have the tight junctions, which is the occluding junction, and then the zonal adherence, zonal occludens, and the macular adherence of desmosome. These make the terminal bars that we can see sometime. Here we see intestinal absorptive cell with the apical end, and these are microvilli projecting on the surface. And here we can see where lipid is actually being resynthesized. So these are not lipid droplets per se, but they are lipid being resynthesized by the smooth endoplasmic reticulum. That's why we see a membrane around it, because that's really to smooth the R. That's resynthesizing the triglycerides that come on as monoglycerides, as we'll talk about later on. So there's basically three things that amplify the surface. One is the pachycircularies, that is the infolding of the submucosa and the amplification of the surface area. That increases by threefold. The villi, intestinal villi, which you only find them in the small intestine, they increase by tenfold. And then the microvilli itself, the brush border here, increased by 20-fold. So the greatest increase in surface area is due to the microvilli at the cellular level. Now, in between the epithelium, you got the lamina propria. So it's connective tissue. Also, there may be some muscle cells running through there. Certainly, you can see the muscle right there, the muscularis mucosa. Also, there's a central lacteal. So there's a lymphatic. You can see the endothelial cells very nicely of these cells, endothelial cells running through there and there of this lacteal. So this is a blind ending lymphatic that comes down from the villi, from the villus down through here to take fluid back through the lymphatics into the bloodstream without going to the liver. And there's also the Meissner's plexus in the submucosa. This is muscularis mucosa, muscularis externa. You have the Meissner's plexus, another plexus of nerves that regulate the contraction of the muscularis mucosa. Out of H&E preparation, again, we can see lots of cells that actually have lots of lymphocytes in through there, connective tissue cells that we see. Also the central lacteal. Here's a central lacteal located here and here. And also there's smooth muscle fibers. You can see these smooth muscle fibers run through there, which actually move the villi somewhat. Here we see the terminal ileum on slide 61. This is the muscularis externa, and then this is actually the luminal surface in through there. We can see Pennant cells at the base of these. So when bacteria slip in through there, the Pennant cells are ready to engage them. So intestinal villus is a connective tissue core with epithelium on its surface, and that epithelium is simple columnar epithelium with absorptive cells and goblet cells and Argentaffin cells. As we know, we have the artery and veins going in and out of here. Also, there's a central lacteal. The lacteal comes out that takes fluid to the lymphatic drainage. There's also lymph nodules that could be in the lamina propria or the submucosa, usually in the submucosa, but could be up there. And also we can see the nerve plexus, the Meissner's plexus, that occurs in the submucosa that interacts with smooth muscle and the various things that are located under nervous control. They are located in the villus. And here we can see the very big granules right at the bottom of these cryptic lubricant are the pennant cells, the muscularis mucosa, in through there, intestinal absorptive cells, and we can see the goblet cells as well, and the central lacteal are right there. The terminal ileum, we can see again intestinal villi projecting in through the muscularis mucosa, submucosa, the epithelium, and the serosa. Here we can see Prior's patches located in through here, and lighting through there maybe would be some of the M cells. Certainly they're in here somewhere close to the lymphoidal tissue that amplifies it presents antigens. These cells present antigen to the lymphocytes that are below. And then here in the submucosa, we have a plexus, as well as the Auerbach's plexus that's located between the intercircular and outer longitudinal muscle of the muscularis externa. So here we see the muscularis externa, the submucosa, and the mucosa with lymphoidal tissues at the edge. So the mucosa would be from here to there. Lymphoidal tissue is going across the muscularis mucosa that we can see lymph nodules lumen in through there. The appendix does not have villi. It only has these intestinal glands, as does the large intestine. Again, we can see the appendix. We can see the lymph nodules around the mucosa in through there, and this is the submucosa muscularis externa. Here again, we can see the real thing. This is the epithelium on the surface. They have goblet cells in through there. Lots of lymphoidal tissue in the appendix. This is the appendix that we're speaking of right here. You have lots of goblet cells, simple columnar epithelium on the surface with goblet cells and absorptive cells. So in the small intestines, you have intestinal villi. It's pretty flat. In the large intestine, you do not have villi, but in both cases, you have intestinal glands. So you do have these intestinal glands in a large intestine as well as a small intestine. So we see these glands projecting on basically somewhat of a flat surface. So we can see this flat surface cut in through there. This is the lumen, and this is the muscularis externa. And most of the cells are goblet cells. There's a little bit about, we mentioned the nerves. The Auerbach's plexus is under the parasympathetic activity. So whenever you want digestion to occur, you want movement of a GI tract, food along the GI tract, is the Auerbach's plexus that's responsible for that. However, the sympathetic inhibits movement of GI tract components. It inhibits the ileocecal sphincter, and it inhibits the internal anal sphincter. So whenever you see a bear, it is not time to defecate or digest. It is time to run. So you close these GI functions down for you be able to move things along. However, when you recover from the fright, the parasympathetic will come back and be involved with moving things along the GI tract with paracelsus. And so here's Auerbach's plexus and the Meissner's plexus. Again, another view of it. Submucosal nerve plexus controls the motility of the muscularis mucosa and the secretion activities of the mucosal glands. The Auerbach's plexus controls the motility of the smooth muscle in the muscularis externa, paracelsus, moving things along the way. And then we get the colon, transverse section that we see. This is the intestinal glands. As we see in through there, there's a muscularis mucosa right there, lymphoidal tissue that you can see, the lymph nodule there. Many goblet cells, so there's lots of goblet cells in through there, slip things along. So as we talked about before, we have intestinal villi, no villi in the large intestines, but we do have the glands. So in the large intestine too, the outer longitudinal muscle is thrown into these bands, tinea coli. And so instead of being all the way around, they're in different bands. Large intestine function and absorption of water electrolytes from the ingestible foodstuffs into the feces is what they do. The large intestinal colon of the monkey we can see right here. So the foodstuff would be going down through there with lots of goblet cells, and these are the intestinal glands, which would compose of absorptive cells with brush border and goblet cells as well. And then that brings us next to the mucous cutaneous junction, where you go from the rectum to the anus, where you have stratified squamous epithelium again, and you have a sphincter located with that. Here we can see the sphincter in through the external sphincter, internal sphincter, and we can see the anus in through there and the rectum. So this is simple columnar epithelium with absorptive goblet cell, and all of a sudden you abruptly change to stratified squamous epithelium that we see right in actually transition between right here, right there between these two, and then you come out with skin. So in the recto-anal junction, as soon as you get outside, you're going to pick up hair follicles and the keratinized stratified squamous epithelium of the epidermis, and then you got the dermis. You got goblet cells. You have the sebaceous glands. These are sebaceous glands here, which dump into the follicles, as you see there. Also, it has, in addition to having these regular sweat glands, it has the apical sweat glands, and that's what we see here, which discharges contents actually in the hair shaft as well. And so this is kind of used for marking, maybe for animals, for marking, but it's discharged in the hair shaft as opposed to the regular sweat gland discharge on the surface of the cell, the surface of the epithelium. So in our digestive tract, there's distinguishing features. You got the mucosa, what type of cell might be there from the esophagus, stomach, small and large intestines. And then the submucosa, maybe there's glands, maybe there's not. Located in through there, there would be glands in the epiglottis, as well, in the esophagus, as well as in the duodenum, the beginning of the small intestines, if you have submucosal glands. What's in the different muscle walls, and also the adventitious. In the very beginning, the top part of the esophagus is firmly attached with the adventitious. Likewise, at the end, the anal canal is firmly attached with the adventitious. However, in between, you can have a serosa, or you can have kind of a combination of the two, where you have mesothelium on the outside, and that facilitates slipping and sliding, as opposed to the beginning and end. You don't want the gut to slide whenever you're moving foodstuff down through there, and so those are anchored down with the adventitious. Now, in the clinical correlation, we've already talked about this. A person had a disease where he had gastric atrophy. You get deterioration of gastric mucosa, and that causes a decrease in parietal cells. Parietal cells produce intrinsic factor. Intrinsic factor is needed for vitamin B12 absorption. If you have vitamin B12 deficiency, it reduces the proliferation of erythroblasts and a decreased red blood cell number, and a person becomes anemic. And so the gastric atrophy is not only the bleeding of the gastric area itself, but also the loss of production of the red blood cells. We want to thank various sources for the original books that have been modified for this presentation. Those were the original sources of these images. This is the end of the digestive system 1. We'll pick up with the digestive system 2, which is associated with associated organs like the liver, the pancreas, the different organs that are associated with the GI tract but are really not part of the tube. And so that's pretty much the end of it. I hope it's useful to you.